If your organism is diploid, a mouse or a pea plant, say, and the mutant phenotype is recessive, there is a simple solution. You breed heterozygous individuals, carrying one good copy of the gene and one defective copy. They will have a normal phenotype, but when they are mated with one another, 25% of their progeny will be homozygous mutants and will show the lethal mutant phenotype, while another 50% of progeny will be heterozygous carriers of the mutation like their parents, allowing you to keep on breeding. But what if your organism is haploid? One method hinges on the use of temperature-sensitive mutants. In these mutants, the protein product of the gene is only conditionally defective. It functions normally within a certain range of temperatures, the permissive temperature range, but can be inactivated by a small shift in temperature that takes it out of this range. See Figure 1926. Thus, the abnormality can be switched on and off experimentally simply by changing the temperature. A cell containing a temperature-sensitive mutation in an essential gene can be kept alive by growing it at the permissive temperature. It can then be driven by a temperature shift to display its mutant phenotype. Figure 1928. Many temperature-sensitive mutants were isolated to identify the genes that encode the bacterial proteins required for DNA replication. Here, large populations of mutagen-treated bacteria were screened for cells that stop making DNA when they are warmed from 30 degrees Celsius to 42 degrees Celsius. Temperature-sensitive mutants have also been used to identify many of the proteins involved in regulating the cell cycle, discussed in Chapter 18, or in moving proteins through the secretory pathways in yeast, discussed in Chapter 15. Genes involved in complex phenotypes, such as changes in learning or behavior, can also be identified by genetic screens in model organisms. As an example, researchers were able to isolate a gene that affects social behavior in worms by screening for animals that feed alone. Figure 1929. A complementation test reveals whether two mutations are in the same gene. A large-scale genetic screen can turn up many different mutants that have the same phenotype. These defects might lie in different genes that function in the same process, or they might represent different mutations in the same gene. How can we distinguish between the two possibilities? If the mutations are recessive, if, for example, they represent a loss of function of a particular gene, a complementation test can reveal whether the mutations fall in the same gene or in different genes. In the simplest type of complementation test, an individual that is homozygous for one recessive mutation, possessing two identical copies of the mutant allele, is mated with an individual that is homozygous for the other mutation. If the two mutations are in the same gene, the offspring will show the mutant phenotype, because they carry only defective copies of the gene in question. See Panel 19.1, page 674. If, in contrast, the mutations fall in different genes, the resulting offspring will show the normal, wild-type phenotype because they will have one normal copy and one mutant copy of each gene. When the normal phenotype is restored in such a test, the sets of alleles inherited from the two parents are said to complement one another. Figure 1930. Complementation testing of mutants identified during genetic screens has revealed, for example, that 5 genes are required for yeast to digest the sugar galactose, that 20 genes are needed for E. coli to build a functional flagellum, and that many hundreds of genes are needed to guide the development of an adult nematode worm from a fertilized egg. Single nucleotide polymorphisms, SNPs, serve as landmarks for genetic mapping. With the recent determination of the complete human genome sequence, we can now study human genetics in a way that was impossible only a few years ago. Using the human genome sequence as a starting point, we can begin to identify directly, through gene sequencing, those DNA differences that distinguish one individual from another. No two humans, with the exception of identical twins, have the same genome. Each of us carries in our genome a set of variations in nucleotide sequence that makes us unique. At some sites in the genome, these variations turn out to be common and relatively harmless, so that there is a high probability that any two people chosen at random will differ at that position. In these cases, where two or more sequence variants coexist in the population and are both common, the variants are called polymorphisms. Some of these polymorphisms correspond to deletions or insertions of large chunks of DNA sequence. But the commonest type of variation is due to the substitution of a single nucleotide. Typically, any two humans differ by about 0.1% in their nucleotide sequences, approximately one nucleotide difference for every 1,000 nucleotides. This translates to about 3 million single nucleotide differences between one person and another. These single nucleotide polymorphisms, or SNPs, figure 1931, can be used as markers for building genetic maps or for conducting searches for mutations that correlate with specific diseases or predispositions to disease. The How We Know panel on page 680 to 681 explains how SNPs are used in a variety of ways for this latter purpose. Mutations that give rise, in a regular, reproducible way, to rare but clearly defined abnormalities, such as albinism or congenital deafness, can often be pinpointed through family studies. Common diseases with more complex causes, such as diabetes or arthritis, call for a different approach. For these conditions, there is no single gene that is all-important. Instead, there are many genes, as well as environmental factors, that affect the risk. But with the help of SNP analysis, it is becoming possible to track these genes down. For this purpose, researchers collect DNA from a large number of people who have the disease, and compare those samples with DNA from a parallel group of people who do not have the disease. Suppose, for example, that a particular allele of a gene creates a heightened risk of a heart attack. People who inherit that allele are likely also to inherit a set of SNPs that are genetically linked to that allele. Thus, SNPs are exceptionally common among heart attack patients. Thus, SNPs that are exceptionally common among heart attack patients serve as flags indicating that a risky allele of a gene lies nearby in the genome. Figure 1932. This approach has been used to search for genes affecting propensity to common human diseases such as diabetes, coronary artery disease, rheumatoid arthritis, bipolar disease, manic depressive illness, and several others. For all of these conditions, environmental as well as genetic factors play an important part. Figure 1935. Moreover, for all of them, the genetic factors themselves are complex. Not one but many genes contribute to the incidence of each disease. By tracking down the genes involved, and the polymorphisms associated with the disease, it becomes possible not only to identify and help those people who run heightened risks, but also to discover more about the molecular mechanisms of the disorders. Linked groups of SNPs define haplotype blocks. Of course, the more SNPs one has to analyze to track down the location of a gene, the more costly and laborious it is the enterprise. Fortunately, for the purposes of genetic mapping, it is not necessary to examine all 3 million SNPs in the human genome. This is because SNPs are not all independent and uncorrelated. They are linked to their nearest neighbors in blocks, called haplotype blocks, that tend to be inherited as a unit. Thus, individuals who carry a particular variant at one polymorphic site are highly likely to carry correlated variants at all of the neighboring polymorphic sites within that haplotype block. Figure 1936. It is enough, therefore, to examine one or two representative SNPs for each haplotype block. To understand why haplotype blocks exist, we need to consider our evolutionary history. It is thought that modern humans expanded from a relatively small population that existed in Africa about 60,000 years ago. Among the small groups of ancestors from whom we are all descended, some individuals will have carried one set of genetic variants, others a different set. The chromosomes of a present-day human represent a shuffled combination of fragments of the chromosomes of different members of this small ancestral group of people. Because only about 2,000 generations separate us from the ancestral population, large segments of these ancestral human chromosomes have passed from parent to child, unbroken by the crossover events that occur during meiosis. Remember, only a few crossovers occur in each human chromosome per meiosis. As a result, certain sets of alleles and DNA markers such as SNPs are inherited in linked groups. These ancestral chromosome segments, sets of alleles and markers that have been inherited in clusters with little genetic rearrangement across the generations, are the haplotype blocks. Like genes and genetic markers, which exist in different allelic forms, haplotype blocks also come in a limited number of variants that are common in the human population, each representing an allele combination passed down from a particular ancestor long ago. Haplotype blocks give clues to our evolutionary history. A detailed examination of haplotype blocks can provide some remarkable insights into the history of human populations. New alleles are continually being generated by mutation. Many of these variants will be selectively neutral. That is, they will not affect the reproductive success of the individual, and so they have a chance of becoming common in the population. The more time that has elapsed since the origin of an allele, the smaller should be the haplotype block that surrounds it, because over the course of many generations, crossover events will have had many chances to separate the allele from the SNPs nearby. In fact, by comparing the size of haplotype blocks from different human populations, it is possible to estimate how many generations have elapsed since the origin of specific mutation. In this way, combining genetic comparisons with archaeology, we can trace our history from that small set of African ancestors and even draw deductions about the most probable routes our ancestors took when they left the continent. Figure 1937. Selectively neutral alleles take a long time to become established in a population, but an allele that is strongly favored by natural selection will sweep through the population and build to a high frequency rapidly. For example, a mutation or variation that makes an organism more resistant to an infection will be favored by selection because organisms with this variation will be more likely to survive and pass the mutation on to their offspring. Again, haplotype analysis can be used to estimate when such historical genetic events occurred. If a favorable mutation cropped up in the population relatively recently, there will have been fewer opportunities for recombination to break up the area around that mutation, so the surrounding haplotype block will be large. Such is the case for two different genes that confer resistance to malaria. The alleles that confer resistance are widespread in African populations, where malaria is rife, and they are embedded in unusually large haplotype blocks, suggesting that these protective variants rose to prominence recently in the African gene pool, probably about 2,500 years ago for one of them and about 6,500 years ago for the other. In this way, analysis of modern human genomes can reveal the ancient history of human exposure to specific infections and of our development of resistance to them. In revealing the paths along which humans evolved, the map of human haplotypes and SNPs provides a new window into our past. In helping us discover the genes that make us susceptible or resistant to disease, the map may also lead us towards new ways of coping with our present medical problems. Essential concepts. Sexual reproduction involves the cyclic alternation of diploid and haploid states. Diploid cells divide by meiosis to form haploid gametes, and the haploid gametes form two and the haploid gametes from two individuals fuse at fertilization to form a new diploid cell. During meiosis, the maternal and paternal chromosomes of a diploid cell are parceled out to gametes so that each gamete receives one copy of each chromosome. Because the assortment of the two members of each chromosome pair occurs at random, many genetically different gametes can be produced from a single individual. Crossing over ensures the proper segregation of homologous chromosomes and enhances the genetic reassortment that occurs during meiosis by exchanging genes between maternal and paternal homologs. Although most of the mechanical features of meiosis are similar to those of mitosis, the behavior of the chromosomes is different. Meiosis produces 4 genetically dissimilar haploid cells by 2 consecutive cell divisions, whereas mitosis produces 2 genetically identical diploid cells by a single cell division. Mendel unraveled the laws of heredity by studying the inheritance of a handful of discrete traits in garden peas. Mendel's law of segregation states that the maternal and paternal alleles for each trait separate from one another during gamete formation and then reunite at random during fertilization. Mendel's law of independent assortment states that during gamete formation, different alleles segregate independently of each other. The behavior of chromosomes during meiosis explains Mendel's laws. If 2 genes are close to each other on a chromosome, they tend to be inherited as a unit. The frequency of recombination between them can be used to construct a genetic map that shows the order of genes on a chromosome. Mutant alleles can be either dominant or recessive. If the heterozygous organism has a mutant phenotype, the mutant allele is dominant. If it has a normal phenotype, the mutant allele is recessive. Complementation tests reveal whether 2 mutations that produce the same phenotype lie in the same gene or in different genes. Mutant organisms can be generated by treating animals with chemicals that damage DNA. Such mutants can then be screened to identify phenotypes of interest and, ultimately, to isolate the responsible genes. With the exception of identical twins, no 2 human genomes are alike. Each of us carries a unique set of polymorphisms, variations in nucleotide sequence, that shapes our individual phenotypes. Polymorphisms are sites of high variability in the genome sequence, such that 2 individuals drawn at random from the population have a high probability of being different at those sites. Single nucleotide polymorphisms, SNPs, are polymorphisms where the variability is in the choice of a single nucleotide, with 2 or more variants being common in the population. SNPs provide useful markers for genetic mapping. Human SNPs tend to be inherited in large haplotype blocks, segments of the genome that have been passed down intact from our distant ancestors and in most individuals have not yet been broken up by meiotic recombination. The sizes of haplotype blocks give clues to our evolutionary history.